All right, thanks, Matt. It's a pleasure to welcome attendees today to the 16th in our networked speaker series for the Center for Dark Energy Biosphere Investigations. We launched this program really to help establish and maintain our collaborative community of researchers, giving opportunity particularly for younger researchers to tell the community about what they're working on and discoveries they've made and questions that they're hoping to address in coming years. It's a particular pleasure today to be able to introduce Dr. Gus Ramirez. I had the pleasure of meeting Gus at sea on an expedition to the Northeast Pacific in 2011, and he was part of the team of researchers and microbiologists who were on board helping to get some of the first high-quality samples that came out of long-term borehole observatories on that expedition. Dr. Ramirez earned his PhD at USC in marine biology and biological oceanography in 2016, and earlier this year he joined the Daunt Lab at the University of Rhode Island on a C-DEBI postdoctoral fellowship. Dr. Ramirez has received numerous awards, including an NSF GRF when he was a grad student, and he's working on a variety of projects in different settings, both in the deep ocean, and he's worked in shallower water as well. And today we're going to hear a presentation on microbial netter quartet, life and death post-entombment. Gus, take us away.
Well, thank you very much for the kind introduction, Andy. And thanks to everyone that's logged in to check this out live. So with that, we'll go ahead and get started. I'd like to begin by showing this slide. This is data published in the Proceedings of the National Academy of Sciences Journal by Kallmeyer and colleagues in 2012. And what we see here is logarithmic cell concentrations as a function of sediment depth for a number of different places in the ocean. All of these places are color-coded, and what we see in panel B is a map depicting where these places are, and also panel B shows sea surface chlorophyll concentration superimposed on the map. Sea surface chlorophyll concentration is usually a great proxy for primary productivity of the water column directly above each one of these sediment sites. So the first point that I want to make here is that the highest cell densities that we see in this global survey come from areas that have extremely high productivity. These areas are close to the continents usually. Inversely, if we look at areas of extremely low productivity, such as the South Pacific Gyre, we can see that there are orders of magnitude less cells at any given depth sample if the waters above the sediment are less productive. The second point that I want to make here is that independently of the density of cells that's found at the sediment interface, for all sites across this global survey, we see exponential declines in cell magnitudes as a function of sediment depth. These inclines are particularly accentuated in more or less a decimeter to a meter of depth beneath the seafloor, and where we see 2.5, almost 3 orders of magnitude decreases in overall cell densities. This, of course, implies that 90 to 99% of cells present at the water-sediment interface die after approximately 10 centimeters of burial. So this has a number of interesting implications for understanding the nature of life beneath the seafloor and subseafloor sediments. The fundamental question that I will try to address here, however, is whether or not we are co-extracting DNA from dead cells when taking total DNA indiscriminately from subseafloor sediments. This is a basic question because we want to make sure that we frame our hypothesis about what we understand in the subseafloor based on information from things that are potentially alive rather than things that are potentially dead. As easy as this concept is, it has not been addressed for a number of reasons, 2 reasons primarily. First and foremost, the concept of prokaryotic viability is not an easily tractable issue. It's really hard to agree, it's really hard to get a large community to agree on when a cell is dead versus alive, especially in a low energy environment. Second of all, there's a limited amount of methods out there that aim to address who's dead and who's alive, particularly in complex environments like subsea floor sediments and terrestrial soils. So what I like to do now is expand on this concept of microbial viability by showing this route to death as published in AEM in 2011. What we see here is basically a gradient of metabolic and cytological changes that have to occur for communities that are mostly alive and considered biomass to transition into communities that are mostly dead and considered necromass. One important thing to keep in mind is that an intact membrane appears to be an absolute requirement for high levels of metabolic activity. And the reason for that is, of course, the Mitchell hypothesis. So the Mitchell hypothesis states that we need intact cellular membranes in order to separate proton gradients that ultimately drive ATPases. So when a membrane is damaged, we lose that proton gradient, and we can no longer power ATPases, regardless of whether or not there's nutrients in the environment. So again, when mostly living communities begin to be challenged by their environments, we may even see metabolic spikes early on. These spikes represent the community trying to address that challenge. However, when the challenge is perpetual, such as is the case in subsea floor sediments, which are energy deprived environments, at some point the metabolism will decrease below detection level, and at around this point as well, we will begin to see cellular damage occurring across the entire community. This damage to the cell membranes can add up to the point where cells simply begin to burst. And when a cell bursts, intracellular contents such as nucleic acids, lipids, carbohydrates, and proteins become part of the extracellular milieu. And when any one of these biomolecules is detected as part of the extracellular milieu, we don't really call them living. We know for a fact that they are simply fractions of cells that were once alive that are no longer alive. And that's what we consider necromass. So to a first order approximation, it is tempting to say that cell membrane integrity is a really good proxy for microbial viability in nature. I agree with this statement, and I've been exploiting this idea by utilizing a molecule called propidium monoacide. And what propidium monoacide is, it is a DNA intercalating dye that intercalates DNA, and if it's exposed to a bright light, it creates a covalent bond between the DNA double helix. DNA that is covalently bonded is unable to denature at 95 degrees, one of the basic steps for polymerase chain reaction amplification. So therefore, it will not amplify after doing PCR. Another thing that I want to point out now is that intact cellular membranes are completely impermeable to propidium monoacide. Therefore, in a natural environment where you have living cells with intact membranes, you have cells with damaged membranes, cells with extensive damaged membrane that we can consider moribund or bound for death, and cells that have been long dead whose DNA is now part of the extracellular environment. If you treat a complex sample like this with PMA, you can selectively amplify 16S genes, in my case, from the living community. That is, you can selectively amplify DNA that was shielded from PMA intercalation by an intact cell membrane of a very potentially viable cell. So by taking subsamples and then treating a subsample with a PMA and then having a non-treated control and comparing differences between these two samples, any sizable difference can be directly attributed to DNA from moribund cells or DNA from long dead cells that is now extracellular in the environment. This is quite a robust technique. It's been used in a in a number of different environmental settings. However, I believe that I'm probably one of the first people to attempt to use this photoactive dye to tell viability in subsea floor sediments. So, excuse me. So to address whether extracellular 16S genes inflate 16S counts reported from indiscriminate total DNA extractions from the environment, and if this inflation may have an effect on high-throughput sequencing conclusions, I embarked on 2 oceanographic expeditions. The first of these expeditions was on board the Norwegian research vessel GeoSARS, and we visited the flanks of the now famous Loki's Castle hydrothermal vent system in the mid-Arctic last summer. Earlier this year, I embarked on a second expedition on board the research vessel Sally Ride, And we visited the borderlands, or the general vicinity in the eastern Pacific around the Mexican and U.S. border. Excuse me. Following all sediment collections at both of the sites, fresh samples were subsampled into PMA-treated and non-treated controls in the field. Both replicates were light-activated in order to form covalent bonds in the DNA double helix, and then frozen for shore-based analysis. Once I was back on land, I was able to extract DNA from all of these samples and then perform quantitative PCR quantification of 16S genes, along with successfully getting amplicons for the V4 hypervariable region of the 16S gene, and sending those amplicons out for sequencing on an Illumina MiSeq platform. So I like to begin by showing quantitative PCR-based enumerations of total versus intracellular bacterial 16S counts. These results aim to address if quantities reported from indiscriminate total DNA extractions are inflated by potentially extracellular genes in nature. These extracellular genes, of course, represent remnants of long-dead cells in the environment. So let's go ahead and take a look at the data. Here I show 3 cores collected from the Eastern Pacific sites. The x-axis depicts 16S rRNA gene loads per cubic centimeter of wet sediment in a logarithmic scale. The y-axis shows sediment depth in centimeters below the seafloor. Total gene counts are plotted in red, and intracellular gene counts are plotted in green. And generally, this data reflects similar trends that we have already seen with subseafloor cell counts. That is, we see extremely high gene counts in the shallow-most sediment, followed by extremely rapid decreases in those counts in the first 10 centimeters or so of sediment, 3 orders of magnitude less for Pacific Core 2 in this case, and then some sort of a stabilization 1 to 2 meters into the sediment. Regarding the influence of extracellular 16S rRNA genes, the main conclusion here is that for all Pacific sites that we sampled, the majority of sample depths show no statistically significant differences between total and intracellular gene counts. Generally, this is good news because there are a number of different studies that have already published total gene counts from indiscriminate extractions of DNA from subseafloor sediments. And what our data suggests is that based on PMA results, our data, along with other reports that may already be part of the literature, are likely to reflect 16S genes from communities that have intact membranes or are most likely viable in the subseafloor. Another thing that I like to point out here is that for 2 cores at 2 depths, 8 centimeters below the seafloor, specifically here in Core 1 and Pacific Core 3, we do see an inflation of total 16S genes reported relative to intracellular 16S genes. Again, this inflation can be directly attributed to a high amount of either dying cells or cells that are long dead, meaning extracellular DNA in the environment. When looking at our Arctic sites, we see almost identical results. That is, extremely high numbers in the shallow-most sediment, followed by rapid decreases in the first 10 centimeters and somewhat of a stabilization. And again, the main story here is that there are no differences for most of the samples that, for most sample depths that we've interrogated. However, for 1 out of these 2 cores, we again see an inflation that can be directly attributable to extracellular 16S genes or genes from dying cells in the shallow-most sample horizon of Arctic Core 2. So to go ahead and summarize this results, I guess that I can still say to some extent that hypothesis 1 is correct. Extracellular 16S genes inflate total 16S gene quantities. However, this inflation is only detectable in shallow sediment. And for the sake of simplicity, when I say shallow sediment, I mean 10 centimeters or less below the seafloor. However, deeper horizons and the majority of shallow sediment depths that we'd sample here too show no significant differences between 16S gene counts reported from total 16S genes versus intracellular 16S genes specifically. So, okay, next I'd like to show results from high throughput sequencing. And I will show sequencing results in 3 parts. The first of 2 parts will be based on broad taxonomic descriptions, basically alignments to the latest rendition of the Silva database that place a taxonomic stamp on the sequences. The third and final part will show the results of standard OTU-based techniques using more sophisticated ecological metrics. So let's go ahead and look at the Pacific data first again. So what this figure shows is the relative phylum level contribution of bacteria and archaea for each of the 3 Pacific cores at all depths as reported by intracellular DNA and total DNA. So again, each one of the depths that you see here has 2 bars reporting the community. One of these bars shows community from intracellular DNA and is labeled as PMA, and the other bar shows communities from total DNA and is simply labeled as DNA. Major community differences here don't appear to be driven by intracellular or total 16S genes reported communities. Major community differences here appear to be simply a function of sample depth. So we can see that at all sites, the 3 Pacific cores, we look at the shallowest depth horizons, they are generally more diverse at the phylum level. We get contributions of both bacteria and archaea, and there's a high number of phyla that comprise the communities. The deeper and deeper you go, for the most part, proteobacteria and firmicutes appear to take over the communities, and to some degree the JS1 candidate division as well. Again, like looking at this data, it seems that the major driver of community is not the amount of extracellular 16S genes, but rather where in the sediment column we've sampled these communities from. So to a first approximation, we can say that differences per sample based on total versus intracellular DNA pools are negligible. Very similar story is it's what we get from looking at the Arctic data here. There's less variability with depth in each one of the 2 cores, but again, generally, both intracellular and extracellular communities depict extremely similar patterns. So, all right, we can draw, I guess, a premature conclusion at this point and say that extracellular genes appear to have little to no influence on reported community compositions at the phylum level of taxonomy, however. Again, this is just the phylum level of taxonomy, so this conclusion can easily be challenged by simply looking at higher taxonomic resolution comparisons, and that's what I'll show next. And to do this, I like to revisit the 3 sample pairs, 2 in the Pacific at 8 centimeters below the seafloor and 1 in the Arctic at 2 centimeters below the seafloor, where we know already, based on 16S qPCR, that extracellular 16S genes exist in those particular depths. So again, we have already looked at these communities from total DNA and intracellular DNA at a broad taxonomic description and have seen that there are no real differences. The phylum level taxonomy composition and distribution at these sites where there are for sure extracellular 16S genes based on qPCR, again, appear unaffected by the presence of those genes. The same story is true at all 3 of these sites. So what I want to do next is show higher taxonomic resolution comparisons based on something called taxonomic heat mapping. And that's what you see here. So I'll go ahead and zoom in on those heat trees and explain them further. So these taxonomic heat trees depict quantitative visualizations of community diversity data in a taxonomic context. That is, the abundance of depicted taxonomic hierarchies is represented by node color and size, and this relationship is summarized in the color scale legend at the bottom right. In essence, these are taxonomy maps for all sequences in each data set that allow tracking of sequences at their highest level of taxonomic assignment. The tree to the left shows the taxonomic high-level taxonomic assignments for all sequences from the intracellular DNA data set. So this is beyond phylum level. You can see families, gamma protobacteria, and the structure in deeper taxonomies there at the branches. The tree to the right shows a similar scheme, identical. Tree to the right shows the taxonomic structure of the community of both intracellular and potentially extracellular 16S genes. And when, again, looking at deeper and deeper taxonomies at the class, order, and family levels, we can conclude that they are both still highly similar. This implies a number of things, but I interpret this as meaning that the pool of extracellular DNA, extracellular 16S genes represents the pool of intracellular DNA genes, meaning that the things that we're most likely to find dead in nature are most likely to be affiliated with the things that are alive. So there's some sort of equilibrium going on here. That's how I'm interpreting this data. Again, a nearly identical story can be drawn from looking at the second Pacific core. Very little differences. Higher taxonomic levels are observed between the two communities. And again, in the Arctic core, the overall shape of the community here is different because it's a different benthic environment. But overall, we can say that there are no major differences at higher taxonomic hierarchies for these two communities. So based on visual inspections of the data at broader taxonomic levels and phylum, it seems as though extracellular genes have no effect on reports of community composition and structure. However, there are more powerful analysis methods out there, of course. And what I've done next is cluster the sequences based on percent similarity at a 97% cutoff into something called operational taxonomic units. So once you have your data in this in OTU format, you're able to use more sophisticated algorithms that aim to parse out differences in a quantitative way between any one community against any other community. So I'll begin by showing a principal coordinate analysis of Bray-Curtis dissimilarity distances for Pacific and Arctic sites. So again, what we have here is that each sample for the Pacific and each sample in the Arctic have been assigned a specific color and plotted in two-dimensional space here. Data from total DNA is plotted as circles, and data from intracellular DNA is plotted as triangles. So there are two ecological stories going on here. So let's begin with the Pacific. What we see in the Pacific is clustering along axis number 1 based on sediment depth. So all shallow samples come together, intermediate samples come together, and deep samples come together. And these clustering patterns that, again, suggest depth-based stratification of communities spread across axis 1, which explains 43% of the variance of the data set. A different ecological story is visible for the Arctic samples. In the Arctic, we don't see clustering based on depth. We see clustering based on site. So core 1's data has negative axis 1 values, while core 2 data has positive axis 1 values. And this clustering explains 27% of the variance of the data set. So again, the take-home message here is that each ecological inference based on ordination here, meaning depth-based clustering in the Pacific and site-based clustering in the Arctic, can be drawn by independently looking at only the circles that are plotted, meaning total DNA data, or only the triangles that are plotted, meaning intracellular DNA data. So overall, we can summarize that. We can summarize these observations by saying that ordination results are not skewed by the presence of extracellular genes in the environment. So here I will take a quick look at alpha diversity metrics. So again, this is data for Pacific Core 1 showing richness, diversity, and evenness metrics. Total DNA data is plotted in red, intracellular DNA data is plotted in green. And just very generally, we can see that the scores are actually quite similar, but even more importantly than the scores, we can see that the patterns across depths mimic each other pretty well too, I guess, with the only differences being evenness scores in deeper and deeper samples. But overall, there appears to be, again, no influence of extracellular DNA genes in richness, diversity, or evenness. So by now, we can conclude that both broad and high resolution community comparisons of total versus intracellular DNA appear to make no difference in reported in results that we report from high throughput sequencing. So therefore, it would be pretty easy now to say that our hypothesis 2 here is debunked and say that extracellular 16S rRNA genes do not influence prokaryotic surveys of subsea floor sediments. However, Billy May says there's more, and hypothesis 2 is the gift that keeps on giving. So what about tracking the influence of extracellular 16S genes on individual OTUs across sediment depths? That's something that I had not done. Anyway, let me expand on this a little bit. Given that we have total and intracellular relative abundances for each OTU in every sample across every depth, we can look at variations of intracellular sequence contribution to total DNA sequence counts per OTU. So at instances where there is little contribution of intracellular DNA to the total DNA pool, we can conclude that a large amount of sequences for this particular taxon arise from dead cells or extracellular DNA in nature. So let me go ahead and show this. This plot shows logarithmic relative sequence abundances for the 60 most prevalent OTUs for each core at each depth in our combined Arctic and Pacific dataset. Again, all of these abundances come from total DNA. I'm going to show the same exact data as collected from intracellular DNA or PMA-treated sample. Here it is. And what I want to do now is introduce this concept of a viability ratio. So again, this viability ratio, or the rationale behind why dividing intracellular DNA by total DNA tells us something about the viability of any particular OTU in any particular core at any particular depth. The rationale behind that is the following. If all 16S genes are intracellular and thus in intact membranes in nature, intracellular DNA counts must be equal or very closely equal to total DNA counts. And therefore, this ratio, viability ratio, will be close to zero, implying that the majority of the community is viable. Conversely, if most 16S rRNA genes for a particular OTU in a particular core at a particular depth are extracellular in nature or are in cells with damaged membranes, the relative contribution of intracellular DNA to the total DNA pool will be much less, and therefore this ratio will deviate from 1 and approach 0. And we can imply with near zero ratios that most of that OTU community in that particular spot, meaning time under burial per core, can be interpreted as mostly dead. So I did this division for each OTU at every depth in every sample and came up with this new heat map. So this blue heat map shows ratios near 1 as deep blue and ratios near 0 as gray. Here we can see the viability history per OTU across burial times. The first major observation here is that most OTUs are mostly viable for most depths across most horizons, which is good because that's I guess that hypothesis is one of the main drivers behind extracting DNA from deep sea sediments in the first place, because we think that they can tell us something about the living biota of this otherwise incredibly hard to study habitat. The second and most interesting observation that I got out of this was that there appears to be drastic fluctuations in viability as a function of burial time for some dominant OTUs here. So what I'll do next is have a closer look at the viability life histories for these 4 high abundance OTUs, OTU 1, 6, and 12. So OTU 1 is a gamma protobacterium, OTU 6 is an atribacterium, OTU 7 is a thalmarchion, and OTU 12 is a member of the candidate phylum OP8 or Aminosinantes. All right, each one of these OTUs gets their own plot now. And the x-axis depicts the viability ratio plotted in the black dotted line as a function of sediment depth. Superimposed on each plot is the sequence abundance in logarithmic scales from total DNA for that same OTU at each one of the plotted depths. So looking at OTU1, we can see that both its sequence abundance and viability increase with burial time, and thus we can almost say that this OTU seems to be happy, certainly healthier, being entombed for long periods of time. So it shows a depth-adapted lifestyle to, I guess we can say that OTU6 shows this as well to a high degree. It seems mostly alive, mostly at depth after hundreds of thousands of years of entombment. Moving on to OTU7 and 12, we see different lifestyles. Here, viability is near 100% for both taxa in shallow sediment or early entombment times. However, both OTUs will experience drastic declines in viability with depth. And this implies that, particularly for OTU7, when we sequence OTU7 at 10 meters below the seafloor, the overall sequence counts don't change much, but the viability of this OTU changes a lot. So we can say that this guy might contribute to the biology that's occurring early on, just a few centimeters below the interface. However, if we kept tracking this guy for an extended period of time, even though its sequences may stay constant, its relative contribution to the community may stay constant, we would actually be tracking more and more dead cells in the environment, and something that's less and less relevant in terms of framing biological hypothesis of what's happening at this depth versus what's happening at that depth. So anyway, I'd like to conclude with 3 main points. The first point is that statistically significant differences in bulk extracellular 16S genes are only detected at relatively shallow depths. Second point is that sequencing-based microbial community surveys appear to be unaffected by the presence of extracellular 16S genes. And the third point is that OTU's specific relative contribution of DNA from dead or dying taxa may vary drastically with sediment depth and time. So this is all I have. Thank you all for listening. I guess we have time to take questions.
Absolutely. Thanks, Gus.
Thank you.
I can't hear all the other applause, but I'm sure there's plenty. If folks would like to, they can enter their questions using the chat window. And I can already see one question there. And then Gus, I'm not sure if you can see that chat window. It might be a good idea to read them out loud just so we have them on the recording for this, and then others can also benefit from that. So you want to go ahead and start with, looks like we have a question from Julie Huber.
Yeah, correct. Yeah. Julie is asking if I have taken a look at finer nucleic acid levels, that is 99% similarity cutoff, I suppose, or oligotyping. The answer to that, Julie, is that I have not. This is definitely in the works. I've been spending most of my time trying to figure out a pipeline for doing a discriminative metagenome, but it certainly would not hurt to look at higher similarity cutoffs and reanalyze this data and see what kind of effect that has on viability ratios for sure. Yeah, that's a really good point. Okay, Stephanie Carr says, Gus, can you comment on the geochemistry at these steps? Yes, very briefly. The samples from the Eastern Pacific and the geochemistry at the Eastern Pacific sites and the geochemistry at the mid-Arctic sites were very, very different, of course. I guess I'll begin just by discussing oxygen. Oxygen was absolutely gone from all sites in the Eastern Pacific at the first millimeter or 2 millimeters of depth. Whereas in the Arctic, oxygen penetrated close to 40 to 50 centimeters sometimes. So yeah, just this difference alone sets up incredibly different geochemical topographies, right, where these communities exist. So I guess that we have already quantified the difference by looking at the taxonomic heat maps and seeing how drastically different communities from the Pacific sites were relative to the Arctic sites. So again, the Arctic is going to be, I guess, an abyssal pelagic benthic habitat, and the water depth at the Pacific sites was close to 1,000 feet. So that would be mesopelagic. Yeah, there's fundamental differences in the geochemistry and physics that set up these communities. So, okay, Jason Sylvan. Did you solve the potential issue of sediments quenching the light required to activate PMA? Yeah, that's PMA can. It's not a perfect solution to what we're after, right? So there's a number of caveats that I'll put up front regarding the application of PMA for figuring out who's alive and who's dead in nature. First of all, I guess that the major issue with PMA is that you can have a sizable fraction of the population have damaged membranes that can potentially recover from that damage. So you would be efficiently silencing things that may be alive at deeper depths by overdoing PMA. Another thing that's an issue with PMA, and one of the main reasons why it hasn't been applied to more complex samples, is because it requires this light fixation step, correct? So, when you have a sediment, it's hard to evenly shine light across a sediment slurry. So that can lead to a number of complications. However, I think that the data that I'm presenting here is still quite robust, and I didn't see light activation as a potential issue when it came to forming covalent bonds in the samples that I presented today.
Now let's see.
Myrna's asking
Looks
if I
like
have
there's
activity.
one more maybe.
Oh, sure, yeah, I'll answer one more. Myrna, hi Myrna. She's asking if I have activity measurements. I do not, and I've talked to Myrna extensively during my tenure at USC about doing intra and extracellular ATP or adenosine assays before. So no, maybe that's something that we can work on, but I haven't done that yet.
All right, looks like we have a little bit more. Let's see if there's one more question might be coming in. Let's see. Oh yeah, so there's a question here about having sediment left over and whether it's worth talking with some other folks about co-locating measurements. That's of course one of the hallmarks of a lot of the C-DEBI projects is trying to measure as many different angles on these problems as possible in the same locations, and I think that's something we all find pretty exciting. So I can imagine you'd be curious to know about what else could be done with some of those samples or from samples collected nearby.
Oh, absolutely, yeah. More than open to collaborate. If people have ideas, I'll be more than happy to hear them, of course, yes.
Hey, here's a naive question, Gus. Because as a non-microbiologist, seeing what you presented, and it was really a really nice talk. It was very well presented, and I think I understand conceptually what's going on here. How new is this approach more broadly? In other words, outside of the deep sea microbial community,
Yeah.
how extensive is this approach being applied to other settings? I'm just curious kind of where it fits in terms of being something that there's a lot of history for it, or it's so novel that there's a lot of work going on just figuring out how to use the tool.
The approach has been around for quite some time. I mean, I came out of the food industry. I worked in the food industry for a while, and I remember people attempting to quantify, the fraction of viable microbes after lighter and lighter pasteurization methods. So that your food would taste the same if you over pasteurize, so you can overcook it. So they tried to do this with PMA. After working in the food microbiology industry, I did some work, some government work. So I was an intern at the Jet Propulsion Lab, and my team there also used this technique in order to try and figure out why there was such a diverse community being collected when swabbing DNA off of spacecraft assembly clean rooms, which are like the cleanest places on Earth. That's where they put together the Mars landers and all that. So it turned out that 90% of the DNA that they were swabbing were simply remnants from once living organisms that had been long dead. And that was the hypothesis going in, that a lot of these things had to be dead because they were spraying the floors with hydrogen peroxide and irradiating them with ultraviolet light 17 hours a day. So this approach is well cited in the literature. It's there's a lot of potential for it. Again, there's always caveats, but it's I'm just glad to be able to bridge this technology that's been around, and again, in industry and in government for quite some time, bridge that into what we do here at C-DEBI.
Interesting, yeah. So it seems like in some ways it's akin to looking at both the DNA and the RNA to try to distinguish between what's there but maybe not active. But this seems like it's a pretty robust approach where maybe you don't have to worry quite so much about, say, that environmental preservation as you would with RNA.
Yeah, correct. And I initially set out to try to find out who was alive based on RNA as well, but we run into problems there too. Because of the extreme low energy flux in subseafloor environments, particularly in diffusion-limited environments like sediment, where metabolic activity just drops below detection level, we may be missing a lot of what's there if we're simply pursuing living communities based on RNA. So this could be a good complementary approach to a lot of other much more established techniques in the field. Yeah.
All right. Thank you. And Jackie Gordial or Gordile notes that PMA is used extensively in permafrost ecosystems to look at DNA preservation. So there's another example that's pretty interesting.
Cool.
Very nice. Well, it looks like we've hit the questions, and we're right on time. So I'm just going to thank Gus once again for participating in the program and telling us about these new exciting results, and we look forward to seeing future work in this and related areas. So thanks, Gus. And I want to thank, you're welcome. You're welcome. I want to thank Matt Janicek for facilitating and managing the AV system, and then all the participants and everyone who's dialed in and seen this network speaker presentation today. I'll just remind everybody that we have the next networked speaker presentation by Dr. Emily Estes from the University of Delaware, and that's going to be in November 2nd, so just a couple of months here, also at 12:30 Pacific Standard Time. So hopefully we'll see a lot of the same people and maybe some more people as well at that presentation. Take care, everybody.
Thank you.
You're welcome.